In this video, we're going to learn about sensory receptors, so how we obtain and process information. We'll look at a few just as a brief overview, but we're going to focus mainly on vision and hearing. Sensory receptors are highly modified ends of sensory neurons, and they can be grouped together to form a sensory organ. And group receptors will amplify the energy of the stimulus to ensure that threshold is achieved, and we send a signal to the central nervous system to be processed so that we can respond. This is a table that's taken from our old Nelson Biology 12 textbook, talking about the different receptors. So taste receptors are a chemical stimulus, so our taste buds identify. Smell is also a chemical stimulus, and it's detected by olfactory cells in our nose. Pressure is a mechanical stimulus. Proprioceptors is also mechanical, and that helps us position our limbs, so we kind of have an idea of where we are within 3D space. Balance is due to our inner ear. It's mechanical, and as body movement causes us to be able to make adjustments and know where we are. Our outer ear detects sound waves. Our eye detects light, so it changes in light intensity. Movement and color are all perceived by the eye. And thermal regulators, the stimulus is heat. So this is a section of the eye. We're going to highlight a few of the main structures and their functions. So one thing you need to be able to do is label and annotate an eye. This is from our Cognitive Higher Level Biology textbook. So the eyelid is not something we have to discuss, but basically it can close over and protect our eye. The sclera, which is found here, it goes all the way around here. It is the white of your eye. The exception is in this part right here. That's the cornea. The sclera protects your inner structures of your eye. The cornea is an extension of the sclera, but instead of being opaque, it's transparent. And it needs to be transparent because it needs to be able to refract light into the pupil and then onwards through your eye. The aqueous humor is right here in this section. This blue, it's the blue section here. Its job is a watery liquid that protects your lens and also gives your cornea the nutrients it needs to stay healthy and alive. The next part that we want to take a look at is the choroid layer. So that's this pink layer, the lighter pink layer. It's the middle layer, and that's where your blood vessels and dark pigments to absorb light and stop reflection are found. Your iris is right here on both sides. That's the colored part of your eye. It's made of circular muscle, and it also controls the size of your pupil, and it monitors the amount of light that enters your eye and opens and closes the pupil or the opening. The lens is right here. Its job is to focus the image, and there are some ciliary muscles or ciliary bodies that are attached to the lens, and they help control the shape of the lens to focus the light properly so that you can see. That doesn't always work so well, and that's why some of us need glasses. The whole inner section here is the vitreous humor, or it's filled with vitreous humor. It's kind of a jelly-like material, and it's found behind your lens, and it helps maintain the shape of your eyeball. Your retina is this dark red or kind of fuchsia section. It is the inside layer of your eye, and that's where we display our images. And it's lined with Light sensitive cells. So our rods and our cones are found there. So rods detect light and dark, and cones detect color. Our bipolar cells are found there. We'll talk about those and how they're involved in seeing. And then your cells from your optic nerve are also found along your retina. The fovea centralis is right here, and for whatever reason, this is the area where your vision is the sharpest. There's a higher collection of rods and cones in that section. This is where your sharpest vision is. Right here, this part of your retina is called your blind spot. So you actually can't see anything there because that's where your optic nerve is. So there's no rods and cones in that spot, so we can't get any information about vision in that spot. So our rods we'll talk about and our cones in our next diagram. Rods, their job, they detect light in black and white, and they're the most numerous light sensitive cells, and they respond to low intensity light. So their job is to basically see in the dark. And that's why when it's dark, you can't see as much color, because cones only operate in bright light. And they help us identify colors, red, blue, and green, because we have blue, we have red, and we have green cones. So those three help us to see the myriad of colors that we can see. So red-ring color blindness, people with that condition, it's more appropriately named red-green color deficiency. They have a gene defect in the photoreceptors in either red or green cone cells. So both of those defective genes are recessive, and they're found on the X chromosome, which means that males are more likely to develop the condition than females, because they need 2 copies. Females need 2 copies of the recessive gene to express it, but because males only get 1 X chromosome and the other one is a Y, they only need 1 copy of the recessive allele. This is a picture of the retina. There's quite a bit of information going on here. Here's the choroid layer here. This is the pigmented epithelium. These are the cones, the green, red, and blue cones. These are rods. This is called a bipolar cell because it synapses with the cones or rods, and also with these ganglion cells that are here. This is the vitreous humor, so light is coming from the lens through this way. So one of the skills you need to be able to do is to identify the direction of light. So it goes through the ganglion cell to the rods and cones to the back of the eye and off to the optic nerve to get processed. So, as I said before, the bipolar cells synapse with both rods and cones and ganglia. When there's no light, the rods and the cones depolarize and send inhibitory signals to the bipolar cell. When light is present, the rods and cones stop sending that inhibitory signal, and that causes the bipolar cell to depolarize, and it activates the ganglion cell. And then the ganglion cell will send a message to the optic nerve to go to be processed in the visual cortex. So the retina is made up of, as it says at the bottom here, pigmented epithelium, plus rods, plus cones, plus bipolar neurons, plus ganglion cells. It's a pretty complicated diagram. You don't have to reproduce it, but you do need to identify the direction that light travels. So one thing you need to be able to know as well is the contralateral processing. So our two eyes help us perceive the size and distance of objects. So information from the right half of your visual field is detected by the left half of the retina in both eyes and is processed by the left hemisphere, and the vice versa for the left half of the visual field. So you can see that in this diagram here, which is also from Cognigy. Let's talk about the ear. So this is a structure of the outer, the middle, and the inner ear, there's quite a lot of structures involved. So your outer ear, the thing that we call our ear, doesn't actually have a huge amount of impact except for funneling sound into the auditory canal. That's called the pinna. So what we know as our ear is called the pinna, and its job is to funnel sound, it kind of acts like a funnel, into the auditory canal. The auditory canal, which you should never stick things into, but people do all the time, conducts sound towards the eardrum, which is also sometimes called the tympanic membrane. And the eardrum vibrates and then sends sound waves into the middle ear. And in the middle ear, there are 3 bones, which are called ossicles. They're really tiny, and they have funny names, and they're kind of named based on their shape. So the first bone is the malleus, which is the hammer, the incus, the anvil, and the stapes, which literally looks like a stirrup off of a saddle. So those 3 bones hit each other because of the tympanic membrane movement, and they conduct and amplify sound and carry it to the oval window, which is found at the end, right at the end of the stapes, or the stirrup part of the bone. If a really loud sound comes through, those bones can actually stop vibrating so that to protect your ear. So now let's take a look. So the oval window, like I mentioned, is here. It's oval shaped, and again, it has a membrane on it. It's a hole at the beginning of the inner ear. The eustachian tube here is an air-filled tube that connects your mouth and nose, that helps equalize pressure between your outer and your inner ear. So when your ears feel plugged, you can try to equalize that pressure by plugging your nose and making a blowing motion. You're actually clearing out your eustachian tubes. Your inner ear is this section here. It's got your vestibule, this part right here, vestibule, and then your cochlea, which is shaped like a snail, and 3 semicircular canals that are all in 3 different planes of motions, like a X, Y, and Z axis. So they basically point in the 3 directions. So the vestibule, like I said, is the chamber right here at the bottom of the semicircular canal. So this is our vestibule, and it gives information about the equilibrium, so about your head position. So there's a couple of reasons why that happens, and we'll discuss it as we continue. So the semicircular canals, as I mentioned, are 3 fluid-filled canals positioned at 3 different angles. And the movement of fluid within those canals gives you information on body movement and your position. So basically, if you were spinning really quickly on a amusement park ride, and you kind of get discombobulated, you don't know which way is up, it's because everything's moving so quickly that your semicircular canals can't perceive which way up is. So that you can feel a little confused for a bit because of that. The cochlea here is shaped like a snail, and it responds to sound waves and converts them to nerve impulses and then sends them off to be perceived as sound. Inside this curled up cochlea are structures called hair cells. Basically, they vibrate and they have different lengths. and those lengths help us hear different pitches. So those hair cells identify different frequencies and intensities of sound. They're very fragile, however. So if intense and prolonged noise above a certain decibel level happens, you can actually break those hair cells off and they don't regenerate. So then that's what causes hearing loss. You actually naturally lose hearing, your range of hearing as you get older. So children and teenagers can hear far larger range of sound than even somebody in their early 20s or 30s or 40s and so on. So you can actually do some fun tests to see what range of sound you can hear. And if in class we did that experiment, I would stop hearing sounds in the higher range of sound than the students in the room, because I am older and I've lost some of the range of the higher pitches, as do all adults. There's actually an interesting case example. I believe it was in Great Britain. Students would loiter outside of like shopping malls and stuff, and they were considered a detriment to customers coming to shop at the mall. So the owners of the plaza decided to project a sound that was very high pitched, and the idea would be that the students wouldn't like the sound, the teenagers would be repelled. I don't think it went over very well, but the older people wouldn't be able to hear the sound because it was outside of their range of hearing. This is a zoomed in version of the inner ear for you to get a better look. So these are the semicircular canals. There's one, and there's another. There's a vestibular nerve, so a nerve that's sending an impulse. And inside of these, you have this, where there's some hair cells. So those hair cells vibrate and send information to the vestibular nerve. So let's discuss how we hear. So first of all, sound waves come in through the auditory canal, hit the eardrum, and cause it to vibrate. The vibrations are passed to the ossicles, those bones, the hammer, the inca, and the stapes, or the stirrup. Their job is to concentrate and amplify those vibrations. They actually triple the force of the sound that's coming in. And then, as I said before, the oval window receives the sound wave and moves inward. And then the round window, which was on the picture of the ear, we'll just go back. There's the round window. It moves outward. So the one moves inward and the other one moves outward. And then those motions trigger movement of fluid inside your inner ear. Your cochlea receives the waves and converts them to electrical impulses, which are then interpreted as sound. We're going to discuss how we hear sound now. So the inner workings of the cochlea. So imagine this is our cochlea, all curled up like a snail. The hearing device in the cochlea is called the organ of Corti. And it's made of one inner row and 3 outer rows of hair cells, which we can't really see here. The hair cells are anchored to what is known as the basilar membrane. So right here. And the hair cells are covered with a gel. So when there are fluid vibrations, they move the basilar membrane. So here we go. And it causes the hair cells to bend, and that movement stimulates a sensory neuron in the basilar membrane, and the auditory information is sent to our temporal lobe by the auditory nerves. So how do we hear pitch and loudness? So the cochlea becomes more and more narrow as it coils, as you can see here, and the hair cells become shorter as that happens too. The narrowest part of the cochlea is sensitive to the high frequency sounds, and the wider part of the cochlea responds more to low frequency sound waves. And each frequency or pitch ends in a separate auditory section of the brain. So that's how we can hear pitch and loudness. So one requirement for you for IB exams is to explain how cochlear implants work. So that's Cognity section A.3.5. But you can also watch a crash course on what does a cochlear implant sound like if you'd rather on YouTube. They help explain it quite well. You definitely don't hear the same way when you have a cochlear implant. And you have to have an external device to help you hear, but then you also have an implant right into your cochlea to help you collect your sounds and interpret them.